I will conclude this session with a discussion of lymphatic endothelial cell biology. We'll have plenty of time for questions, and if we exhaust those, we'll all get a little break before we head on to the clinical part of the program. So, lymphatic endothelial cell biology, just like the blood vasculature, the lymphatic endothelial cell is a major orchestrator of what the lymphatics are capable of accomplishing. And their biology is unique. It differs from that of the vascular endothelial cell, that is the blood vascular endothelial cell. And what I'd like to do is give you a little bit of an overview about this very special cell so you have a context in which to think about the clinical aspects later. So I think it is illustrated fairly well in this schematic. We've touched on some of this, the fact that the blood vasculature, in essence, is the source of the preload on the lymphatic vasculature. And in addition to providing fluid to prime this pump, there are also macromolecules, the antigen-presenting cells and dendritic cells, and lymphocytes that we've talked about, as well as in certain circulations, the absorption of lipids. Again, remember, one of the central structural differences between a blood vessel and a lymphatic vessel, the blood vessel has these pericytes that define some of the vascular biology of the blood vasculature. Lymphatic vessels do not have pericytes, but in contrast, they have these very unique structures of so-called anchoring filaments that actually attach the lymphatic endothelial cells of the initial lymphatic capillaries to the surrounding interstitium and are probably central to the way in which the vasculature is able to work as an uptake mechanism. This is what lymphatic endothelial cells look like under the microscope. I don't have any special message here except for you to have a graphic image of the cell type that we're currently talking about. When we look under standard light microscopy, it's exceedingly apparent that there are differences between arteries, veins, and lymphatics. So first you can see the much smaller relative size of this arteriole. This is a venule, which has, of course, a thicker wall, and this is a lymphatic, which you can readily see is composed of a single cell layer. The cell type under light microscopy looks quite similar to what you see in the venule, but you can see the distinct differences in structure. Under slightly higher magnification, again, you are looking at isolated lymphatic endothelial cells in the lining of this lymphatic capillary, and again, you can see the substantial differences in structure to the arteriole and the venule. Most importantly, the presence of red blood cells in these vascular structures, which of course are absent or should be absent in large measure in the lymphatic capillary. So schematically, you can see depicted on this slide the various attributes of the lymphatic vasculature as orchestrated by the lymphatic endothelial cell. We're going to divide our consideration among the initial lymphatic, which is the other term that is used for a lymphatic capillary, the pre-collecting lymphatic, the collecting lymphatic. So the initial lymphatic, which you see in cross-section here and more in tubular structure here, is composed of blind-ended sacs. They are highly permeable to interstitial fluid, to immune cells, and under the wrong pathological circumstances to tumor cells as well. They have thin, discontinuous walls, and there are no supporting cells in the form of pericytes, as I already mentioned. Here you see the same structure in cross-section, and there is some basement membrane material that is present on the lymphatic capillary, but it is discontinuous to allow that convection of fluid that's necessary into the lumen of the capillary. And this is in contradistinction to a blood capillary, which has a well-formed continuous basement membrane as well as the pericyte that I've now said 3 times, sorry. As these lymphatic one more point about the lymphatic capillaries is that they are characterized by a very unique form of intercellular junction that is again discontinuous. This has been described as a kind of an overlapping oak leaf, so that the flaps that are formed in between the tight junctions are able to open and allow again very rapid convection of fluid into the lumen of the capillary. By the time you get to the precollectors and the collectors, these so-called buttons are no longer present. So we talk about buttons and zippers. The endothelial cells themselves interact with buttons that can be open and closed, and the zippers are present on the collecting or conducting vasculature, so composed entirely of tight junctions. So functionally speaking, at the level of the endothelial cell, we want to encourage movement of fluid that will be driven by physical forces, but once the fluid is within the lumen of the vessel, we don't want it to escape again. So we don't want those buttons to be present higher up in the vasculature. What we do want higher up, as Dr. Xavier talked about, are these functional valvular structures which begin to appear in the pre-collecting vessels, and then the very important smooth muscle cell coverage, which again begins to become apparent in the pre-collectors and is fully invested by the time we get to the level of the collecting vasculature itself. And then all of this will transit up to the lymph node and will exit the other side as an efferent collecting vessel that has basically an identical appearance. Well, where do these lymphatic endothelial cells come from? At the very beginning, we're talking about mesodermally derived cellular material that will progress to an angioblast form under the influence of a variety of molecular signalers, and eventually under the influence of vascular endothelial growth factor, these will further derive into endothelial cells. Now we know at this level of development, early on in embryonic life, we have a division between those that are predicated to line arteries and those that are predicated to line veins. And basically that differentiation under the master regulation of COOP-TF2 eventually influences whether Notch is expressed or not. In the presence of Notch, the arterial endothelial cells are able to develop, but in this axis of the development, because of the influence of Notch, there is a suppressive effect that allows the venous endothelial cell to develop. Now we're at the level at which the lymphatic vasculature can arise, and in the mouse at least, on embryonic day, roughly 9.5, under the influence of a master regulator called PROX1, and again there are upstream regulators for it, there is a polarity that begins to be expressed in the embryonic vein such that one wall of that vein is able to progress and develop into lymphatic endothelial cells, which will then modulate the development of the lymphatic vasculature itself. So that process you see right here. So under the influence of PROX1, there is commitment to the lymphatic endothelial cell line through the action of VEGF-C and other growth factor ligands that are specific to the lymphatic vasculature, interacting predominantly with VEGF-R3, which is relatively unique to the lymphatic endothelium. We will see the development of this arborizing specific lymphatic vasculature, and of course an interaction with the development of the lymph nodes themselves. Here's a little better representation of that buttons and zipper structure that I alluded to earlier. So you can see at the blind-ended sac end of the lymphatic vasculature, they are these button-like junctions that are in fact characterized by specific molecular species that are present in the intercellular junctions that allow for these flaps to exist, and that will allow not only diffusion of water and electrolytes, but also rapid convection of fluid and macromolecules into the lymphatic lumen. This is a better representation of those early steps in lymphatic development, again derived from mouse studies in which the anterior cardinal vein begins to show some polarity based on PROX1 expression, which in turn renders these cells sensitive to VEGF-C and allows them to migrate to form the tubes that will eventually develop into the lymphatic network. At the end of this story, because lymphatic endothelial cells, in fact, are different than blood vascular endothelial cells, they have different surface markers that largely reflect the differential in function between this kind of endothelial cell and a blood vascular endothelial cell. So here you can see during the 1990s predominantly, as we began to learn about these various molecular markers one by one, we began to learn which are expressed on both endothelial cell subtypes and which are uniquely expressed or not expressed on lymphatic endothelial cells. And this was a major step forward in our ability to study lymphatic biology in the basic context, both in vitro and from tissues derived from in vivo studies. So here is an example of the way in which we can use these markers in an immunohistochemical environment. I want to specifically point out the differences between ephrin B2, which is expressed throughout the lymphatic vasculature, and ephrin B4, which is more predominantly localized to the lymphatic capillary network. So this is one way we can distinguish not only whether lymphatic vasculature is present, but specifically what part of the lymphatic tree we're looking at within given tissues. LE&RN and potoplanin are both elements that are expressed again on various aspects of the lymphatic tree. Alpha smooth muscle actin is a marker for the smooth muscle component, of course. So it's going to be present on lymphatic vasculature from the collecting vessel above, but not be present in the lymphatic precollectors or perhaps marginally present in the lymphatic precollectors and absent in the lymphatic capillaries. This becomes important, all of this kind of consideration becomes important in the clinical arena, and we'll talk about some of this tomorrow when we talk about lymphatic vascular malformations. But as you can see under the light microscope, a blood vascular lymphatic malformation, like a venous malformation, and which is down below, and a lymphatic malformation up above look exceedingly similar. And a pathologist would have a great deal of difficulty differentiating these two. But when we apply a specific stain, in this case an antibody against D240, which is basically looking for potoplanin, the presence of potoplanin on the endothelial surface, you can see that these vascular structures are potoplanin positive and therefore are lymphatic in nature. These are potoplanin negative and therefore are blood vascular in nature. So this is a blood vascular malformation, this is a lymphatic malformation. Perhaps one of the most useful lymphatic endothelial markers that we have is LE&RN. LE&RN is a CD44 receptor that is present on endothelial cells, and it's a kind of a cannibalization of a receptor so that it performs two functions. One is that it is specifically a receptor for this breakdown product of glycoprotein, which is present in the interstitium, called hyaluronan or hyaluronic acid. Hyaluronic acid flux out of the interstitium is dependent on the normal function of CD44 receptors. And it is apparent that in states of disease like lymphedema, you're going to have impaired hyaluronan breakdown and transport out of the interstitium, which is one of the attributes of the disease, and it's because of the diminished density of these functioning receptors in the interaction with the tissue. But these receptors are also receptors for immunocytes, and it's a very interesting example about how the function of the lymphatic endothelium is redundant or overlapping among multiple features, so we can both clear glycoprotein from the interstitium and help these antigen-presenting cells and leukocytes to enter the lymphatic vasculature and perform their immune function. So here's an example under the microscope of a dendritic cell entering the dermal lymphatics in a mouse ear, and we can surmise that this in fact is occurring through interaction with this very specialized CD44 receptor. I just want to show you how we use this particular staining. This is from my own work in an experimental model of murine lymphedema, and here we're looking at a cross-section of skin. Now, there is a lot of non-specific staining of the keratinocytes by this anti-LIVE-1 antibody, but I want you to pay attention to these very large, irregular, and numerous vascular structures that are present in the dermis that are clearly LIVE-1 positive. You see the brown stain that surrounds the white lacune that is the lymphatic vessel itself. So this is very characteristic of human and experimental lymphedema, which is namely an increase in the size and number of dermal lymphatics when there is an obstructive lesion up above. So that concludes our presentations. We've given you an overview of edema formation, of the contractile behavior of the lymphatic vasculature, and of the master regulator, in some sense, the lymphatic endothelial cell, all of which is meant to create a backdrop for our discussion of clinical issues in the session to follow. But we'll have time, plenty of time for questions if there are any. I'll invite Dr. Zavieja to come back to the stage, and each of us will be happy to answer any questions that you have.
David, you use for your research mesenteric lymphatic vessels. Do they really represent, or do you know that they represent, their physiology is the same as the soft tissue lymphatic vessels?
So we wondered that same thing. So a number of years ago, we did an analysis of, so if we look at the same type of lymphatic, so if you look at a collector that's in the same relative part of the lymphatic tree in that particular tissue, we looked at them in the mesenteric, we looked at them in the soft tissue of the lower leg, in the soft tissue of the upper leg, and in the neck, as well as the thoracic duct. And then we compared this type of contractile pumping activity amongst those different tissues, because they're under different hydrodynamic conditions and different tissue conditions when the animal is moving or not moving, or eating or not eating. And what we found is there are some underlying similarities that are pretty strong. So in tissues, in all of the tissues, they have both this tonic contractile ability, like blood vessels do, and they have this phasic ability, even in the vessels of the thoracic duct, which we did not expect. Now, the conditions under which those vessels exert that type of activity and how strong it is varies dramatically. And to give you generalizations, if we looked further up into the vascular tree, the lymphatic vascular tree, and got more peripheral, so if we went to the hind limb, for instance, or the lower leg, those were the vessels that had the most and strongest pumping activity. They still had the tonic activity, though it was not as prevalent as it was in the bigger vessels. And as you got to the more central vessels, whether they're up in the chest or in the neck, those vessels had very weak phasic pumping activity, but they still had it. They were less sensitive to things like stretch or pressure that just modestly stimulated this activity, but they were much more sensitive to things related to flow. So as you increase flow, you increase shear in them, and whatever little pumping activity they had, they would shut down. So there is great differences in heterogeneity. The basic principles, though, are actually the same. And if you look at soft tissue lymphatics, if you look at a popliteal or a leg or an arm lymphatic as an example of that, they actually exhibited more powerful lymphatic contractile activity, pumping ability, than those in the gut.
That's actually very interesting, because in my clinical practice, we do a lot of lymphangiogram. And we barely see the contraction, and I think you asked me this question, we barely can see the contraction of muscular or soft tissue vessels, but we clearly can see contraction of the thoracic duct. So I guess the anatomy and physiology of mice or small rodent and human are different.
So mice, I think you're correct that mice and humans are quite different, because actually mice, the pumping activity in mice is significantly lower than most of what I showed you today. Most of what I showed you today, 90% of that was done in rats or larger animals. If we look at mice, which we've done, there's only certain strains of mice and only certain parts of the lymphatic bed that exhibit the same phasic pumping activity. To be honest with you, we're not 100% sure why. I believe it's a matter that size really does make a difference here, because you have to, in a larger, an animal, for instance, a rat just looks like a big mouse, right? It's about 10 times bigger, so the distances involved are about 10 times longer. And we believe that the rat has strong pumping activity because it needs it. The mouse is small enough that the extrinsic pumps, like in the intestine, there is no phasic pumping activity in the mouse. We've never, ever been able to find it. Done hundreds of studies. Every animal above that size, from rat, guinea pig, hamster, dog, cat, pig, cow, and porcine, ovine, human, all have it. So we think that you're correct in that you have to be careful about how you compare results from some of those, but you have to be particularly careful how you compare this type of activity from mouse to much larger animals. And in fact, we know that as you go to humans, their pumping ability is much more potent than what we see in rodent models, for instance. So there is some concern. The curious thing about the thoracic duct is we haven't been able to study that one in situ. We haven't figured out how to be able to get a microscope lens in there and image it without disrupting the whole dynamics of the chest. And so we can't study them in situ like we can all the other vessels that I talked about, the neck, the arm, the leg, the gut. We can study those all in situ on an anesthetized basis, but the thoracic duct we can't. So we really don't know what those changes look like dynamically in the animal with a closed chest. I've always wondered, and particularly after we talked right before the session, whether the changes you're seeing are actually not necessarily the contractile pumping activity of the thoracic duct, but the interaction of the vessel being exposed to external forces of the pleural pressure changes throughout respiratory cycle.
We actually measure routinely a pressure in thoracic duct, and it absolutely has its own contractility. We do approximately 1 or 2 kits, cardiac kits a week. So, and almost every time we get inside the thoracic duct, we measure. We didn't publish this data, but no doubt that thoracic duct has its own contractility. The other aspect is the amount of flow, because the flow in the thoracic, 80% of the flow in thoracic duct originates in liver lymphatics and the intestinal lymphatics. And I'm sure it's changed somehow the structure of the wall, because it's much more higher pressure, much stronger, much faster flow. Can you comment on that? Did you study this?
So a little bit, because we actually thought about that quite a lot. And what we know is that the thoracic duct is one of those that is we consider it one of the weaker of the vessels we've studied in the body in terms of pumping, phasic pumping ability. And it's one of those that is most sensitive to shear rate. So in the thoracic duct, if we would put a 0.5 centimeter gradient of pressure across the vessel that we isolate, all phasic pumping activity would stop in 90% of the vessels. And in fact, if we looked at the different parts of the thoracic duct, if we look at that closest to the diaphragm, it almost never has intrinsic phasing pumping activity. If we go more higher up in the thoracic cavity, that's where we would typically see the vessels that have intrinsic phasic pumping activity. Now, that can change when you expose it to different mediators. So,
sorry, it's all still in rats, right?
Yeah, this is still in rats, and that we have not done in larger mammals, so I can't comment on that. We've tried it in rats. We've tried it in mouse, and again, they simply don't have phasic pumping activity in the thoracic duct that we have been able to discern.
Can I ask how important is the phasic intrathoracic pressure change to specifically thoracic duct flow?
Yeah, so since it's difficult for us to do the types of microscopic flow measurements you'd need there, it's really tough to answer, to be honest with you, but our feeling is that the thoracic pleural pressure changes in at least these rodent animals are the predominant driving force for the extrinsic lymph pump in the chest. There has been someone that studied that in rabbits in Italy and shown that's in fact the case, that there still can be this intrinsic pumping activity, but the thoracic pump, if you will, from breathing rates is the one that predominantly drives it in a slightly larger rodent, the rabbit.
I'd like to ask a question on behalf of the clinicians in the room, including myself, which is, the question often arises because we so frequently see edema as a complication of calcium channel antagonism. What is known about the effect of those drugs on lymphatic contractility? And then part B of the question would be, speaking in general, is there any prospect or horizon for pharmacology specifically to alter lymphatic contractility for diseased individuals?
So I'll answer the first question, because it's easier. We know that nifedipine, trinitridine, all the classic L-type calcium channel antagonists in the rodent vessels we studied, all the rodent vessels we studied, will shut the phasic activity down cold. It appears to block the calcium entry that you need related to the electrical depolarization that occurs from the action potential spreading from muscle cell to muscle cell. And if you block that small calcium entry, you don't get the burst of calcium release from the sarcoplasmic reticular stores. And it's that latter part that actually drives the phasic pumping, the striated muscle type of contraction, if you will. The L-type channels are also important for tone generation. So just like you would use them in blood pressure for control, you actually diminish lymphatic vascular tone, which is very low to begin with, but you diminish it even further. So I've had, as we talked about earlier this week, had lots of clinicians come and ask us about this work, and in fact, it's very, very clear that the calcium channel blockers will have a high potential for impeding the phasic lymphatic pumping mechanism. It's very dose dependent, and so I don't know how it correlates to what you would see clinically in your patients being treated for blood pressure. That unfortunately I can't attest to. Secondly, are there opportunities for developing lymphatic specific pharmacological agents to either increase or decrease pumping? We hope so. We haven't been able to identify them specifically yet. Everything that we've identified will also either affect, because of its similarities to arterial muscle, it'll affect vascular smooth muscle, sometimes in ways you don't want it to, or it can affect cardiac muscle because it acts as a heart as well. So you have this problem we haven't found any that are specific one way or another. Now, there are some unique characteristics of the lymphatic muscle that we've identified. They're very, very potent, and a very, very potent activator is substance P. We don't quite know why yet. We discovered it a while back with some work from a colleague of ours. And it's the most potent vasoconstrictor that anyone has been able to identify so far in lymphatic muscle. We suspect it's because of the interaction and their importance in their role of being immune cell modulated that we'll talk about tomorrow, but in fact we don't know that. We also know that because of this unique dual nature of striated and smooth, that there may be some opportunities for finding agents that work specifically on lymphatic muscle. And one of those that we're looking at are some of the troponins that regulate the phasic muscle activity. They exhibit 2 of the 3 classic cardiac troponins. They don't exhibit one of them. And so we don't know exactly what's taken the place of troponin I yet, but there is a troponin I-like protein that we can identify in the proteome of these cells, but it appears to be a little bit different than the skeletal or cardiac isoforms.
Please.
Hi there. You were talking about intrinsic and extrinsic pumping action. What do you think of decongestive lymphatic therapy? Do you find that truly makes a difference on the extrinsic pumping or intrinsic contractility of the lymphangion?
So we know for sure from work one of my mentors did many years ago, that extrinsically, things of that nature, not just specifically of decompression therapy, but also other types of therapies, and including things like exercise and activity, that will greatly stimulate the extrinsic pump. And so, for instance, there was work done in the 70s where they looked at lymph flow out of the hind limb of a dog, and they could exercise it through electrical activation of the muscle cells and show that lymph flow can easily go up 50 to 100-fold from the skeletal muscle of this dog. They could also passively move it in a similar fashion as you would see in terms of the changes in the parts of the leg, and see not quite the same level of lymph flow increase, but very, very large lymph flow increases. So for certain, those types of activities will have a very profound effect on what we call the extrinsic pump. And in actuality, what you're making is an additional non-normal extrinsic pumping mechanism. The question that we've wondered about is, what does it do to the intrinsic pump? We think it can actually potentially activate that. We have not done those studies per se, but in doing the types of activities we're talking about, you can in essence move lymph flow from the initial lymphatic capillaries and the pre-collector beds, which is a substantial volume of the lymphatic tree, to those places where you have the muscularized lymphatics and have intrinsic pumping. And in essence, you increase preload to them. We know for a fact that preload is one of the most classic potent activators of the intrinsic pump. And so we believe that by doing those types of activities, that's in effect how you can be stimulating the intrinsic pump. Though we have not gone into, we haven't, although there's a group up in North Texas that's at osteopathic school that has been looking at that and showed that in fact it does do that very activity.
One very interesting way of addressing that would be to actually look at the difference between cutaneous stretch a la manual lymphatic massage or drainage. We know drainage is the wrong term, but manual lymphatic massage versus pneumatic compression, which would be two vastly different forms of fluid movement and entry, potentially into the lymphatic capillary bed. That would be a very interesting approach.
Yeah, it would be, in fact. We've been approached by a couple times by companies that want us to do that, and we haven't had time yet to be able to do those types of studies. And to my knowledge, they haven't been done yet.
Other questions, comments, objections? Well, we've earned ourselves an extra 30 minutes of break time, so you either have a chance to go to another session that you missed or just take a nice long break. Please, everybody, come back for the clinical session. We hope we have a lot to offer there as well. Thank you.